When physical microarray images are quantified, the glowing fluorescent scan shatters into thousands of unlogged, highly variable digital data points. Limma uses empirical Bayes linear models to calculate differential expression across thousands of genes simultaneously. This modeling approach relies on a specific mathematical state, variance homogeneity. Raw two-color microarray reads fail this requirement. They are inherently biased and exhibit high variability, particularly when expression intensities are low. This density plot shows the raw intensity channels before any analysis begins. Note the widespread discrepancy and the lack of alignment between the green and red curves across different arrays. Standard statistical tests assume these distributions are comparable. Without alignment, a model often mistakes technical dye bias for biological change, leading to false positives. Before we can run linear models through LM-FIT, we must stabilize the data matrix using Limma's mathematical preprocessing pipeline. The validity of biological discoveries in this pipeline rests on the exact sequence used to transform raw intensities into normalized log contrasts. The pipeline begins by mapping the physical and structural layout of the experiment. The targets frame maps the experimental metadata, assigning specific RNA targets, like the ApoA1 knockout or wild-type samples, to the Cy3 and Cy5 channels. These text annotations are merged with the unlogged raw intensities to build an RGList object, the primary storage container for the data. The software uses a spot types file to link specific ID patterns to designated control categories, such as calibration spots or blanks. Image-derived spot quality weights are then integrated as the first line of statistical defense. This scatter plot shows how those categories and weights map onto a real experiment, highlighting control points like sensitivity, buffer, and ratio controls across the matrix. A quality weight of 0 neutralizes a damaged spot's influence on normalization without deleting the data point from the record. Conversely, assigning a weight greater than 1 increases a control spot's leverage, which helps to anchor the baseline. These weights act as a modulation layer, allowing the model to selectively trust data before mathematical transformations begin. Many pre-processing workflows assume that subtracting local background noise directly from the foreground signal improves the data. The intent is to strip away technical interference to reveal the biological signal beneath. However, this plot shows the result of simple subtraction. Note the massive wedge shape and severe fanning of data points at the low end of the intensity axis. Over 1,000 data points are missing from this distribution because simple subtraction resulted in negative intensities. On a log scale, negative or 0 intensities result in infinite log ratios or missing data. These infinite log ratios dominate the variance profile of the experiment, masking the actual biological trends. Naive background subtraction limits the statistical power available for low expression genes, requiring a more precise mathematical approach. Limma addresses this through adaptive background correction using the normexpect method. This is the same dataset treated with normexpect. The wedge shape is gone, replaced by a tight, uniform distribution of the main data cluster. With noise contained, the spike in ratio controls now separate distinctly from the central cluster. Adaptive correction strictly enforces positive intensities, ensuring all data points remain mathematically viable. By adding an offset value to the equation, the algorithm damps the variation of log ratios near 0. This forces the variance to remain consistent across the entire intensity spectrum. Normexpect with an offset provides the mathematical stability that allows weak biological signals to be distinguished from technical noise. Next, the pipeline must correct for spatial and intensity-dependent structural biases across the array. This is the role of within-array normalization, converting the RGList into an MA list of log ratios and average intensities. Intensity-dependent bias is corrected using locally weighted scatterplot smoothing, or LOES. The algorithm recalculates the values to remove the curve, pulling the data onto a horizontal zero line. This process assumes that the vast majority of probes on the array are not differentially expressed. The lowest STIT is robust enough to tolerate up to 30% asymmetric differential expression without losing accuracy. For platforms like Agilent that lack print tip groups, the pipeline applies global lowest instead. Specialized boutique arrays with high levels of differential expression require the analyst to upweight specific, known non-differentially expressed control spots. These normalization steps force systematically biased arrays into a comparable state, completing the data preparation. The accuracy of the downstream linear model depends on these preprocessing steps. We have transformed a chaotic physical scan into an organized mathematical structure. We mapped the physical targets into the raw RG list. We protected low-intensity signals with NormExp and removed intensity-dependent bias with LOIS. The result is an optimized matrix where the linear model can fit cleanly over the data. Variance in this MALIST is now independent of expression intensity. This stabilized matrix allows for empirical Bayes smoothing, providing the statistical power needed to distinguish true differential expression from technical noise.